Hey, this is the video for Biotechnology.3. So go ahead and set up a new entry in your Cornell notes. Remember to start a new page and check out the essential vocabulary. Today we will be discussing plasmid and recombinant DNA. So a plasmid is simply a circular strand of DNA that's found in bacteria. And the reason that we're using bacterial cells is because they reproduce relatively rapidly and they're very easy to work with. So we will use bacterial cells and these plasmid DNA in order to produce insulin. Remember that insulin is a protein. It is a hormone that attaches to receptors on our cells and allows our cells to take in glucose. And sometimes people with diabetes aren't able to produce their own insulin. And it's really expensive to get insulin from a human, so a lot of times we're tricking bacteria into making it for us. And I'm going to describe that process right now. So what we will do is we will use restriction enzymes to cut that plasmid outside of the bacterial cell. Then we'll use different restriction enzymes in order to cut that plasmid. Once it is opened up, we will insert a strand of DNA that makes human insulin. So this part is from a bacterial cell, and this part right here is from a human cell. So this is the gene that produces insulin. And we will insert that into the bacterial plasmid, and this will be placed back into the bacterial cell. Now this DNA strand is called recombinant DNA. And if you look at the word recombinant, it kind of looks like recombined. And it is a combination of DNA from two different organisms. In this case, it is DNA from bacteria and DNA from a human. Then what happens is we are tricking this bacteria because DNA, remember, are the instructions to make proteins. So this bacteria is going to keep making proteins regardless of whatever DNA is inside of it. So we're basically tricking this bacteria into making these proteins, and this could represent the insulin protein. So this bacteria is just going to mass produce this insulin, and we can obtain this insulin inside a laboratory, and we can isolate this insulin and then use it for diabetic patients. So just to review, what we will do to produce insulin is we will use restriction enzymes in order to cut that plasmid DNA. We will take that human insulin gene and we'll splice it into the plasmid. Then the recombinant DNA is inserted back into the bacterial cell. And finally, the bacterial cells will produce human insulin proteins. Now, genetic engineering is some of the pros. It's good because it has guaranteed results. We can also use it to treat genetic disorders. So somebody might have a genetic disorder and we can insert a gene that might help to cure them of that disorder. And the other thing we can do is produce new agricultural crops. We can make some crops resistant to certain insects. We can make some of them maybe more drought tolerant and different characteristics like that. Some of the cons, it is very expensive. And also, since it's a relatively new process, the long-term impacts aren't really understood fully. And this may cause a lot of people to be a little skeptical and to be cautious of the entire process. Now, what I would like you to do is respond to this check for understanding. I hope that this video was informative. I thank you for your attention, and I will see you soon.